more smooth in the beginning.
Thank you, Volkan. And we will pass to the next one, is comra.
It's over.
Good morning. Good morning. Shi on behalf of comra, I will give you a talk about the Meiofauna study carried out by comra. This is my outline, and since our workshop is the standardization, so I will talk more detailed on how we process our samples. First, I think it should date back to 1996, when China formulated and completed an environmental research program. We call it NOVABA. It's a natural variability of baseline research. The main goal for NOVABA is to understand the natural variability of deep sea ecosystems and its mechanisms. It focuses on the following 4 topics. The first, we try to find the environmental baseline of this area, and the second, we try to find to what extent of natural variability of other ocean ecosystem caused by some oceanographic process such as El Nino and La Nina events. And the third, we try to find the impacts of lateral derived ocean water such as Antarctic bottom water on benthic ecosystems. And finally, we try to coupling the benthic ecosystem with our bottom ecosystems. For these 6, we start our cruise in 1997 in the CCFJ areas, and now we got a contrast area. It includes 2 parts, the west part and the east part. And in the recent year, we also quite interested in some area such as the North Pacific seamount, and we have some cruise on some seamount in the North Pacific. We especially concerned about the bathymetric distribution patterns of the benthos in the seamount. And we also have some cruise in the southwest Indian ridges. And in this area, we interested in the Ghent Meiofauna community. But in this presentation, I would mainly talk about some things we found in the CCFJ area. And this is the sample data we submitted to ISA, including 16 stations, the abundance data, and some data about the species list of nematodes. And we have 3 research verticals to carry out our cruise, including the 大洋1号, 海洋6号, and 向红9号. And we sampled our Meiofauna with TV guide multi-corner to minimize the disturbance of our samples. And after sampling, we got 3 calls for each station. 1 for morphology, 1 for molecular analysis, and 1 for analysis of environmental parameters. We section as follows: 0 to 1, 1 to 2, 2 to 4, and 4 to 6. Not followed the recommendation of the ISA. And for morphology analysis, we preserved in formalin, and for molecular analysis, we preserve our samples in DAS solution. And when the sample was transferred to our lab, we extract from Meiofauna samples based on the Ludok's rotation method. Generally speaking, the sample was first transferred to a series of 6 and And then the remaining sediment was soluted in the noodle solution. And then we stained it with low spangle, and finally in a stereo microscope. For morphology identification, the nematode samples we transfer to a holder which contain 19% fresh water and 5% ethanol and 5% glycerol. We let the samples evaporate slowly about 2 months to let the nematodes emerge in the pure glycerols. And then we mounted it on the glycerin and paraffin wax slides, and we used the DIC microscope to identify the nematodes. 3 reference was listed above, as mentioned yesterday. And for molecular analysis, we generally, according to the ISA handbook, it's the Naturopathic Natural Molecular Protocol handbook. And with the slight difference about the probes, the 4 probes. Here is some preliminary result. We including 3 parts: the Meiofauna assembly structure, the spatial distribution, and some result of interannual evaluations. First, Meiofauna assembly structure. We totally find 22 Meiofauna assemblies from 62 stations, within which nematode dominated and account for about 90% of the abundance, and it was followed by Hypacticoida. And we also tried to find is there any relationships between Meiofauna assemblages? But actually we failed to find. There's no tight interactions with different groups of Meiofaunas. And we studied the nematode biodiversity and finally found that Monstera dominated the community, and followed by Glomadorida, and then it was Anopolida. And totally 151 genus was found in the CCFT in our contrast area, and it was belonged to 8 orders and 44 families. This is some common genus we found in our contact areas, including the Acantholaemus, Alalaemus, and Desmoclex. And we also use the molecular tool to assist identification of nematodes. And we found it was more useful in the Anopolida orders, but it was inadequate in other orders. And we also made an estimation of the genus number in our area. It was estimated that it was about 215 genus may exist in our area. And by examining the buccal cavity, we saw the feeding types of the nematode in our area. Results showing that the field feeding nematode dominated in this area. And the ages we found that more than 15% is in juvenile stages. The second result with the spatial distribution, we found that the eastern part of our contact area, the abundance was significantly higher in the east part, while it was significantly lower in the west part. And we found the nematode variability in the east part and the west part. By cluster analysis and MDS analysis, we found that there was no significant nematode community was found between the 2 areas in genus level, not in species level. And we're trying to find, is there any depth-related distribution patterns in this area of Meiofauna? But no significant correlation was found. So in this area, there may be no depth related. Oh, I'm sorry. Vertical distribution, as mentioned, as documented in other papers, 0 to 1 centimeters dominate the vertical distribution. And the interannual variation we found from 1998 and 1999 to 1970. And we found it was lowest in 2005 years. And we also found some changes of the dominant species in 1999 and 2011. Well, the dominant species in both years was Acantholaemus, and it was followed by Hylolemus, Menegonema, Linhistera, and Monstera in the 1999. Well, when it came to 2011, the subdominant genus may turn to Thalassomonstera, and some genus, just like Comra and Desmocolex, was appeared in the dominant genus. And later we made a cluster analysis of the 2 years, and we found that the 2 years was grouped separated, so indicating that there may be changes of nematode community structure after 20 years. Summaries. We try to find the baseline of our area. And for Meiofauna assemblies, we estimated that it was 22 growths, and for the abundance baseline, that may be 48 to 19 for the western area, and it was 105 to 169 for the eastern part of our area. And for nematodes, it was estimated to about 215 genus in this area. And we also find there's changes during the past 12 years old. Well, we are still facing some problems. Some morphological taxonomic predicament. That is one of our problems, is species level identification. Just as I mentioned previously, we only identify in the genus level, but for species level, we fail to identify. And for molecular problems, and we found that the database, it is better for Anapolida, but for the other orders of the database, it was inadequate. And there's problems about the sequence parameters. Actually, we have tried to more than 10 types of sequence parameter, but none of them was better. And the third problem we face is about the molecular and morphology confliction. Maybe it was found by many labs. So maybe we should try to have a deep thought that we should fill in the gaps. We should take a try to think that to what extent should we do for the environmental assessment? Should we identify in species level? Is that enough for the environmental assessment, or we can improve the molecular problems and molecular tools for the environmental assessments? And finally, I want to introduce some training course organized by the comra. The second workshop on systematic and sampling method held in Hong Kong University of Science and Technology. In the last month, it was focused on the mollusca and crustacean. The goal was mainly on the building the capacity and in the deep sea fauna systematic for purpose of better marine biodiversity resource management and environmental impact assessment. And another training course was held in Shanya in SSE. It's about the deep sea creature. We invited 3 teachers in the last month. Professor Chen, Professor Ross. We only welcome some Meiofauna professor can join our training course. Thanks, the organizer and all experts. Thanks.
Some question?
We have a picture of Joan Bernhardt there. Did you learn about forams from Joan Bernhardt?
No, it was not. I didn't come to.
It was just a start. The final slide.
The last one.
Yeah, the final slide.
Joanne. Yeah.
Yeah. So you have a picture of Joan Bernhardt, who is a specialist on forams. So I was just wondering if you were learning how to identify forams.
I'm sorry, I can't.
Can I help to address this?
Yeah.
Because we realize that for this group, the organism, we don't have very good experts in China for deep sea organism. We have lots of the people working on the fresh water and shallow waters. So we invite the Joanne to give the seminars, and actually more than seminar. She basically talking about how to collect the sample, how to preserve the sample, how to processing the sample in terms both molecular and also the morphological aspect. And then Greg Rose primarily talking about when you have the data, building the data metrics, and how are we going to to analyze the data and building the trees. And there are different methods to building the tree, and talking about the principle and also practical. So we actually taking the students. Well, trainees, I should not call them the students. They're also the juniors, the research scientists. So that we basically show the people how to analyze the data and how to process it. And then when develop the trees and what kind of tree consider as the acceptable tree, what kind of tree consider as an unstable tree. So we using all the actually, we download all the software. We bought this the on-site license. So we download all the software for the tree building, for data analysis, for the sequencing alignment, and all those things basically. In Hong Kong, it's much longer because we have 2 weeks. But for Sanya, it's a very condensed version. So that we only focus on the first part, tree building and the alignment of the data and the data metrics.
Yeah, I just want to say congratulations for your presentation.
Yeah.
I'm very happy to see this, that you are doing a lot with the Meiofauna in the Chinese area.
Yeah.
And especially, I think it's very important the data you showed about the time series.
Yeah.
With this natural variation between the years, because this is really something that we need to study also in other areas in order to understand the natural dynamics.
Yes. So we also try to couple to the El Nino or La Nina events.
The Nino event, okay. And I just want. just a last comment. I forgot to mention in my presentation that we are preparing a special volume in Marine Biodiversity Journal about the biodiversity in the CCZ. So I will send, I think the emails are somewhere in the Dropbox, so you can find the emails of the participants here, so I will send to you this announcement in case you want to submit your data.
Thank you.
Daniel?
It's a really interesting talk, thank you.
Thank you.
You mentioned at the end that you had issues with the species level identification.
Yeah.
That was a problem because there were no descriptions, or did you consider identifying morphospecies A, B, C, or what was the...
ABC, just as you mentioned.
So is that something that you've done or are thinking about doing in the future?
I've done a little and will carry out in the future.
Yeah, this is actually area we're trying to, when we talk to when I talk to the comra, I thought that this is the area we have to strengthen, and so we basically we have run 2 training workshops. First one was on the echinoderm, primarily focused on echinoderm, and second one was on the mollusca and arthropods. We hope to, so we can train our young scientists in this area too.
Anne and Edward?
But I also want to congratulate you. I think you presented some very nice data, as mentioned before, for the nematodes. What I would like to mention here, and maybe some of the molecular biologists could help us, we have similar problems. Success rates of enoplids is higher than of other nematodes. And especially, I don't know if this is also the case for shallow water nematodes, but in the deep sea, we failed to sequence many taxa. Enoplids is not a problem, but for the other taxa, it's a bit unpredictable. So we're still struggling with that, and I don't know if that is a general phenomenon, but I was happy to see that you mentioned it as well. And I remember that Holly Bick in one of her papers focused on enoplids, and we guessed it was actually the same reason. Enoplids went well and all the rest. Yeah.
And I just want to ask, are you sequence used meta-barcoding or a single nematodes to sequence, or?
I'm in the first place talking about the individual barcoding.
Okay.
Elba?
Actually, my comment is related to that I think it is a very good idea to identify it as A, B, and C if you don't have the taxonomists to identify. But it will allow you with photographs, link that to molecular biology, and then maybe go back and do the morphology, which is good.
Yeah, thank you.
Yes, first of all, a really good talk, well presented, I thought, as well. And some interesting data, and it shows a very good progression in the work of comra, I think, in the biological work, particularly recently. My question is to do with the differences that you may show between west and east, and the fact that it didn't show changes necessarily to depth, but of course there are many environmental variables, and a multivariate approach it would be better. So I'm wondering what other factors you might look at, say for instance the proximity to large features like seamounts can affect the distribution of various types of faunal parts. Also, you do have sort of local topography of troughs and ridges, and where you take your samples relative to troughs and ridges may also affect the distribution and the abundance of Meiofauna. So I'm wondering what you think maybe the other factors might be, and how you might address that for the future.
Okay, thank you. This is what I was considering. Take the samples in seamounts, for examples. We have some samples in the foot of the seamounts and in the top of the seamounts, and it was total difference. I think it was mainly caused by the food availability.
I was thinking more in in the comra nodule area, very deep, because of the physical oceanography around large features, that can affect the distribution of Meiofauna within the very deep sediments. It may be a reason why you have differences east to west because of the abundance of seamounts in that region.
Another reasons may cause that.
我们也没测海。
有流产的数据，但是没在那个结合区那边放。
是那个没测。
曾经有过，但是没有一个对比。
Yeah, I guess possibly good idea to go back to look at the topography and some of the hydrodynamic, because they do have some data on the hydrodynamics in this sampling region, and see how the hydrodynamics affect the distribution patterns.
And I think the most important factor may be the food availability.
Yeah.
Right.
Thank you for an excellent talk. One of your first slides showed the photos of your Meiofauna. That was excellent. Just part. Okay.
And the first photo.
Yeah, this one.
Yeah.
Oh, it's fantastic, and I like that kind of research where you all can see what you have found. In fact, you have two very unique animals. The one is, I think, the second species of Spineloricus found in the deep sea, and the tardigrade down there with the head down, I think, at least is a new species, if it's not, it's a new family. You know, if you have some extraordinary on this photo. So are they kept? Are they on your excellent slide you showed? Are they still on the slide?
I'm still preserved in the family.
Oh, well, that's good.
I think you've got a bio there.
Yeah.
And you have to go to the level.
Yeah, that's really great, and I like that kind of research when you take photos.
I didn't bring it this time, and I will go back and find the samples. Thank you.
Thank you.
Okay. Daniel, sorry.
Oh, I'm sorry.
Just have a quick question about your sampling design and how you came up with it. You've got east and west area.
Yeah.
And how did you decide where to sample?
It is hard for me to answer you. How to design the
It's
stations.
a hard question for everybody, I think, to figure out where to sample and why.
Yes. Actually, we sampled in.
I thought they were great, right? Early days, if I remember, there was something great, and then we decided, to transact lines where we're going to collect.
Yeah.
So, I just remember early days I saw possibly almost 10 years ago, when I was in the presentation. I remember there was a map showing to the sea floors, and they have the great design.
Yeah.
And then so along to so they have the grade, and then they have the transact. So this is the sample actually collect, basically in coupling both the grade and transact. Yeah, in both areas.
So transects.
Yeah.
And now that you have more environmental data, do you find that you would like to go and sample somewhere else? Yeah.
Last question.
Okay, could you tell something more about the problems with the primers? They
Primer,
affect to all genera, or do you find some problems related with some of the genera only?
Actually, I have got some hints. Actually, I tried more than 10 primers, and some primers this sequence and compare to the database, the result we found it was fungi.
We know that.
And something we compared, it was found in freshwater samples and not marine.
Okay. That was
Yes, sure.
Yes.
I think this is a big problem, and just because of the database was not well constructed.
More than that, sometimes when you extract the DNA, may have a contamination as well.
Yeah.
Yeah.
Yeah, you have to.
But we have tried. If we failed one time, and we'll try another.
You have to extract the DNA into the biological whole.
Yes.
Okay, thank you very much.